For this module 7.3, we're gonna talk now today about dynamic compartments and how you can use that to model more dynamic cells that quickly adapt to their immediate environment, and how can model complex process like planar cell polarity, which we're gonna learn about soon, if you don't know what it is. This talk will be given by me and Abisha, my student from NC State. So I will start the talk, hand over to her to talk about PCP and a little bit results, and then you go about doing the models, right? How you can use dynamic compartments, and then how you can build your own planar cell polarity model, okay? So remember, this lecture is being recorded and live streamed to YouTube, so you can always go back and watch it again later. If you have, feel free to use all the Zoom buttons like yes, no, go slow, goes faster. If you have questions, don't be shy, send you a message in the chat, and someone from the instructional team will be able to help you, okay? So, yesterday we talked about the difference between structural versus functional way to represent compartments in cells. Okay, so we did a model of cell migration where we had the front layer of the cell, the lamellipodia, the cytoplasm, and the nucleus lagging behind. And then we did another model where you use compartments to separate the cells into a more abstract domains, which will be the apical, lateral, and the basal domains of epithelial cell. And then you later use that in conjunction with links between cells to model apical constriction and for apical fur formation, which is something that you see during the development of many multicellular organisms, right? So everything is fine if the use of those type of compartments. And when you use that, you can, I already said, you can model some migration. You can divide the compartments. The cell automatically take care of replicating the pattern of the compartments, spatial distribution over space as the cells divide. You can use this to also model apical basal an epithelium with apical basal sites, and this is an epithelium that can actually become more longer or shorter. Okay, so you can actually use links between the cell compartments, so between the apical, the lateral, and the basal, and use that to stretch our cell or to compress our cell and make them more squamous, for example. And the example that you did in class, I'm not sure if this movie plays, it's not playing, is where you have the same into the apical compartments, and then you have the subset of the cells constrict the apical compartments, which leads to the furrow formation, okay? And this move here is just to show that you can use those compartments in also as in three-dimensional simulations, right? So this is just a section of an epithelium. You don't see the rest of the epithelium here, but you have this kind of, this will be a process that people call convergence thinning, because the cells converge, but from all the sides, and they do so by preserving their cell volume and getting a taller in one direction. So this convergence thinning process that also is seen in development. Okay, so but in all this process, what's happened is that, okay, the compartments can change shape, they can change directions, right? But they are fixed in the cells. So in this, and by saying I fix that, they are not free to change size of the cell at will. If I just take one of these cells here, for example, and I flip them over, okay, I make the apical side to be this side and the basal side to be on the top side, that cell will have a very hard time flipping it to the correct side to adjust its position, right? You will not be able to do that very easily in the situations, and because those those compartments are fixed in space, and you have to rotate the whole cell for this to happen. And maybe if you're using very small cells, it would be actually maybe a little bit easier. But if you're using bigger cells with more ladder sites, as in the case for this three-dimensional simulation, the cell to flip itself will take a long time. And I may argue that in real life, the cell will not flip completely over if it finds itself for some weird reason upside down epithelium. What will likely more happen is that it will redistribute its internal organelles and signals so that the apical side becomes the basal side and the basal side becomes the apical side. So that's what's more likely to happen. It's not cell completely flipped. So then this seems that the used compartments are not very powerful, if you say like this, but there is a way to use dynamic compartments in Compartmental 3D, which and the trick is basically to make the cells in the that belong to the compartment more fluid and more prone to mixing, and let the external additional energies dictate where each of those compartments should go. Okay, so in a nutshell, that's that's the idea, and this is something that has well, it's not being formally announced in any paper, but it has already been used in previous papers that in CompCell and use our cellular plots model. So one such paper is this one from 2016, where we modeled a section of a nephron, a tube in your kidney, where your cells have a apical, basolateral, and also intracellular domain. And those domains are very dynamic; they can quickly change, and this allows you to have not only to have your tube, but to have the cells that make up this nephron tube to grow and divide, and don't have to worry about placing the compartment in the correct place. The compartments self-sort, so they are to the correct sides. So it detects that there's a lumen inside, there's an extracellular matrix outside, and then they just do that automatically. And with this, you can now not only make a tube that is growing, but you can also model a 6-cell here that grows, divides, and makes cysts of multiple cysts or big cysts or small cysts, depending on all the parameter values. Okay? Another way that you can use cell compartments is, actually I have a movie here where you can see that the cell is growing, it divides, right? And as it divides, you see that the apical side always find apical size and the basal side always automatically sorts out the correct place, because the cell reorganizes its internal environment in response to external cues. So this is something that you can do CompCell and you can use, and it's very powerful because it makes the management of your cells during simulation easier. Okay, you don't have to track where the compartments are and where you should put them. You just place them randomly inside the cell, and the additional energies work in your favor. Okay. So here's another movie where you have the cells that initially they are non-polarized and then they become polarized by apical, basal, and lateral compartments, and then you have links between the external compartments that makes the cells elongate. And here I have add a set of rules that make the cell preferentially form this kind of epithelial balls between the cells, and the cells undergo a series of repolarization events, okay, which will be really hard to, where the cells lost their polarization, repolarize again, okay, if they find themselves in a bad place in the placement in the tissue. And this can only be done if you use the dynamic compartments in your simulations. If you use static compartments, this simulation will never be able to be done to begin with. And finally, another way that you can use dynamic compartments where your compartments self-sort according to the environment is to model planar cell polarity, where planar cell polarity is where you have a tissue that's already polarized, So you have an epithelia, and then the top of the epithelia, where you have the apical side, you have a new set of molecules that sits there, and they can sort out according to the environment and form a polarization in the plane of the epithelium. Therefore, that's what's called planar cell polarity. And this is something that you'll be actually doing at the end of this module. You'll be doing this kind of simulations, and you also be making vector fields to visualize them. I know that you already learned about that a little bit in the morning in module 7.1. Today, we're going to use those vector fields again to visualize the polarization of those cells in your own PCP simulation. So is there any questions in the chat? Well, if there's not, I will switch over to Abisha so she can talk more about what PCP is, what's the state of the field, and what you can do in CompCell to model this. So what goes into PCP, modeling in CompCell 3D. And then after her mini presentation, I get back and we start making models to get a hold of how the dynamic compartments happen in CompCell, and then do the planar cell polarity models. Okay. So I'm gonna stop sharing and let Abisha take over.
Okay. Is my screen visible?
Yes, looks good.
Okay, cool. So I'm going to be talking about subcellular PODS model for planar cell polarity establishment. We know that cells are not homogeneous bags of fluids. Most cells have a heterogeneous structure, spatial differences in shape and structure and function. This is referred to as cell polarity. A classic example of a polarized cell are epithelial cells with apical basal polarity, with apical membrane facing outside surface of the body or the lumen of internal cavities, and the basal membrane orienting away from the lumen or outer body surface. These cells also exhibit something called planar cell polarity, where specific proteins called PCP proteins localize to apical junctions and distribute themselves asymmetrically to the opposite poles of a cell. This polarization of epithelial cells are orthogonal to the apical basal axis, and hence it's called planar cell polarity. This is mostly found in external structures such as body hairs, sensory hairs, in both vertebrate and invertebrate epithelia. So cells orient themselves and their neighbors by establishing asymmetric expression of PCP proteins on neighboring cells. The proteins involved are as shown here. So there's flamingo, Van Gogh, prickle, and all these proteins here. So the PCP pathway finds a fascinating example in Drosophila wing hair orientation, as you can see here. So now I'm going to explain the pathway in brief. So the core proteins, as shown here, form intercellular complexes at the molecular level, and local feedback interactions stabilize these complexes of the same orientation. So flamingo is supposed to localize to both sides, and it helps in cell adhesion. The proteins Van Gogh and prickle gets localized to the proximal site, and Dishevel, Diego, and frizzled gets localized to the distal end of a cell. So in Drosophila wing, this leads to the formation of wing hair from the distal end of each cell. So these hexagonally packed cells at a tissue level align their orientation along the proximal distal axis with the help of some global cues. So various models have been proposed to explain the mechanisms behind planar cell polarity, and these models aim to clarify how the intricate interactions among PCP proteins lead to the observed orientation patterns. Understanding the mechanisms of PCP involves various modeling approaches. There are models that consider molecular level processes explicitly. So these models delve into the intricate molecular processes, analyzing protein interactions and subcellular localization quantitatively. Then there are a second set of models which are called phenomenological or reduced models that capture essential dynamical features of PCB. So these models capture the coarse-grained behavior of the system, highlighting the core principles that drive PCB. So I'm going to give a few examples of some of the important models. So here is an example of an explicit signaling model. This is a reaction diffusion based model and simulation that fully reproduce PCB phenotypes. The geometry in this model is hexagonally packed cells that are fixed in the lattice, and movement of proteins and complexes within each cell has been modeled as diffusion. Now, there are feedback loops arising from interactions of PCB proteins within and between cells, which may be sufficient for the local alignment of complexes between small groups of cells. However, presence of a global biasing signal is required to align the orientation along proximal distal axis. Now, a second example of a phenomenological model is this is inspired by the conceptual resemblance to patterning of electron spins in a ferromagnet or Ising type lattice model. This obeys a short-range consensus rule to simulate what seems to be a self-organized process. So basically, you start with a set of randomly oriented vectors, and the orientation of each vector is modified by adding to it the vector sum of its 18 closest neighbors after that sum has been scaled to 2% of its magnitude. So this is a simple cellular automata model in which a local consensus rule acts iteratively to bias each hair's orientation in favor of the average orientation of its neighbors. So this model describes two systems for hair orientation with the presence of a global bias and a boundary signal. So this is an experimental and theoretical work where the mechanisms that globally orient planar polarity are investigated in Drosophila wing using a combination of experiment and theory. So according to the authors, the proximal distal orientation of PCB complexes does not arise from a random initial configuration, but rather it evolved from a global polarity pattern present at pre-pupil to pupil transition. So at pupil stages, the wing hinge contracts and the wing blade elongates along the proximal distal axis. So this exerts mechanical stress that leads to cell flow patterns that aid polarization. So polarity that was already established when tissue was small can be propagated during growth. So the authors claim that there is no need of a boundary signal to act over long distances directly. So based on these experimental results, they tried to describe this using a vertex model based on attractive interactions between proximal and distal PCB domains across cell boundaries and repulsive interactions within cells. So basically they have an energy term where the energy function where the first two terms represent the PCB interaction parameters from the signaling pathway, and the third term represents the coupling of PCB to cell elongation. So cells were allowed to grow, divide, elongate in this vertex model simulations. So they found that oriented cell division along with cell elongation reorient PCB along proximal distal axis. So in summary, only a few models attempt to describe the reaction between PCB molecules, and all subsequent models are simplifications. And most models use some sort of tissue gradient or a persistent global signal to achieve polarization. And only a few models investigated processes like cell growth and division, whereas most models were static, and most models doesn't have a metric for global polarization. So we are going to use Compartmental 3D to model PCB. So the question is, why do you need a CC3 based model? So first thing, you can use dynamic compartments in Compartmental 3D. It's easy to implement cellular processes like cell growth and division and cell shape changes like elongation and cell rearrangements and all the other features that you've learned so far in this workshop. So using dynamic compartments in CC3D, a cell can be modeled as a collection of objects these compartments can represent either the physical structure of a cell like nucleus or cytoplasm or cell membrane, or abstract regions inside a cell like apical, basal and lateral sides of an epithelial cell. So we are going to use these dynamic compartments to represent the internal distribution of PCB proteins inside a cell. So in our model, each cell has 4 domains: cytoplasm, proximal, distal, and lateral compartments. So proximal compartment represents the proteins that are localized to the proximal end of cell, and distal compartment represents the proteins that are localized to the distal end of cells. So this is how the model looks like. At time t equal to 0, we start from a configuration where all the domains or compartments are completely mixed inside the cell. As time evolves, the system sort out by itself and organize as shown here. So the orientation field vectors point from proximal to distal domains. So the question is, how does the system organize like shown above? Using addition energies. So there were domains that are randomly distributed and the system self-organizes using addition energies. So first we're going to talk about internal contact energies. So the addition energy between internal domains should generally be lower than between compartments of different cells. Only then these you need low addition energy between internal compartments. Now, the addition between proximal and distal compartments should be higher so that they are localized to opposite poles of the cell. So we need high proximal distal addition energy inside the cell so that these two compartments are at the opposite poles. Now, external contact energies. So addition between proximal and distal compartments of adjacent cells should be higher so that these two compartments would come in contact with each other. Whereas the addition between proximal and distal should be lower. So the energy should be higher. And the energy between cytoplasm and other compartments should be the highest. So these are the energies that we are using. Clearly, the internal contact energy between proximal and distal compartments are the highest, and the external energy between cytoplasm and other compartments are the highest. The external energy between proximal and distal compartments are the lowest. So the question is, how did we arrive at these optimal set of contact energies? So we did extensive parameter optimization. So we varied each of the contact energies individually and found the optimal parameter set. So we set the total time evolution of the system to be 1 million Monte Carlo steps. And since the system is stochastic, we took average over multiple simulations to arrive at a degree of polarization that is optimal. So how should we quantify the optimal behavior or degree of tissue polarization of the system? So we need a metric to do that. So the metric that we used was actually the order parameter used by the flocking model community to describe the collective movement of self-propelled particles. It's basically the average of normalized distance between the center of mass of proximal and distal compartments of all the cells in the lattice. So to do parameter optimization, we examined 3 different configurations of the system. So first lattice was a periodic boundary lattice where there are equal number of cells along both X and Y directions with periodic boundary conditions along both these directions. The second configuration had, again, equal number of cells along both directions, but there is a presence of a boundary signal acting from the proximal side. The third configuration, again, had a boundary signal acting from proximal end, but we fixed 4 cells along X direction. So in all these 3 configurations, the total number of cells are kept to be constant. So after this, we computed the global polarization metric, the angle of polarization, and orientation field vectors at every time step. We found the average of the metric over 100 simulations and found success fraction, which is the number of simulations that successfully polarized to 90%. And the average time required to achieve this degree of polarization. Then we compared each metric for 3 different configurations, and that's how we arrived at the optimal set of parameters or contact energies for this model. Along with sensitivity analysis, we tried to understand correlations between different parameters, and we found that the proximal distal, both internal and external contact energies are crucial in driving polarization of the system. Other contact energies like proximal, lateral, distal, etc. are crucial in determining if the pattern is PCP or not. So we found that there exists a threshold value for lateral energy above which the patterns obtained are not PCP-like. Below the threshold, the patterns are more PCP-like when the proximal energy is higher and lateral energy isn't too low. So if lateral energy is very low, we can see that there are domains, lateral domains that get clumped up and the model looks very different from PCP. So once we optimized the system, we increased the system size for 3 different configurations to understand how degree of polarization changes. So first we investigated by increasing cells along both directions with a periodic boundary. The second configuration was increasing cells along both directions with a boundary signal on the proximal end. And third was a configuration with again boundary signal, but just 4 cells along X direction and increasing height with a boundary signal. So we increased the number of cells until 900. So when we increased cells along both directions with the periodic boundary, we found that there is no preferred direction for the system to self-organize. So every time we found different directions preferred by the system. So from the success fraction, we found that as we increase the system size, the success fraction roughly stabilized to 80% when in the presence of a periodic boundary. But now, if we increase cells along both directions with a boundary signal present on the left side, you can clearly see that there is a preferred direction of orientation for the system. All the orientation vectors point from proximal to distal end. So we found the success fraction for this case and found that as we increase the system size, the success fraction decreases and it's less than 70% compared to periodic boundary. This is lesser. Now, we repeated this by fixing 4 cells along X direction and increasing height with a boundary signal and found that almost all simulations were successful. There were 100% success rate, even when we increased the number of cells. So we compared the success fraction for all 3 configurations and found that as we increase system size, the success fraction for the periodic boundary case is higher than with the left boundary. So this is in contrast to what we expect. We believe that there is more freedom in the periodic boundary case when compared to configuration with left boundary signal. And we also found that there is 100% success rate for 4 cells along X direction, which is not surprising because all these cells are closer to the boundary. Now, using this, by fixing 4 cells along X direction, we implemented cell growth and division using mitosis steppable in CC3D, and we try to understand how the system polarizes. So we found that we started the simulation by placing 4 cells along the x-axis with a boundary signal on the left side, and we allowed the cells that are in contact with the medium to undergo growth and division. So the growth rate was kept exceptionally small, allowing ample time for the newly divided cells to orient themselves alongside the neighboring cells. So remarkably, we've observed that the overall polarization remained relatively stable and did not decrease significantly over time. So that's it.
So this simulation, the last one, shows the things that you can do in CompCell, that you not only can model PCP, and you don't actually need to add any fancy stuff in your simulation to do so. You can model with first all the very basic things you already have and you have learned this workshop, but you can, on top of that, have the advantage that you can do you can add all the features that you already learned before, like at cell growth and other cell processes on top of that, and make a simulation which gives a added a layer of flexibility or simulations, right? So you can do more stuff. So what people have done with PCP models before, using code that's not shareable, that you cannot check the code, you can do that in Compartmental 3D, which can actually share the code and people can check what you have done. But you also have the advantage that you can add all these other features on top of it, like in the simulation that Abisha just shown, that you can easily add cell growth on top of it. And you can add more things. You can add cells, some kind of external movement to the cells. So on a field, a vector field, you can add SBML models on top of it, Boolean models on top of it, chemical gradients. All these things you can also do on top of it. So you can not only have a PCP model, but you can have PCP model with other features happening around it, either using PCP signaling and the polarization of PCP as a point of start to do something else, or investigate what other processes can help or hinder the signaling and tissue polarization on planar cell polarity. Okay, Pete had a question on the chat. What's motivated by this study? This study starts just a way to formalize the use of dynamic compartments in Compartmental 3D and make a methods paper about it. But Bish is doing so much things on it that maybe we're going to create make a PCP paper out of it, because we can. She's finding interesting stuff, because there's people say that you don't need a boundary signal, or some people claim that the boundary signal is not enough to ensure global polarization. And then, so I think you're kind of discovering interesting enough stuff that this maybe actually becomes a science paper on top of it. But we started very humble in our objectives when we start this, investigating this. Okay. Okay, and the plan today is for you to make your own PCP model. Okay, so let's now do your series of exercises. Okay, so what you want to do now is to start creating a dynamic cluster. So here you see 4 pictures of cells that are used those dynamic compartments concept, where the cell has compartments, is a cluster of many domains, and you can see that these domains by themselves, they like to mix inside the cell, so they can represent some kind of a molecule or a collection of molecules inside the cytoplasm of the cell or near the membrane of the cell. Okay, and the idea is to choose the contact energies both internally and externally, so then when the cells start to interact with the medium, they start to sort out these proteins or this group of proteins. Okay, so when I say proteins and molecules, I'm making a generic sense. We are not modeling in the individual molecule, we are modeling some kind of a swarm of molecules that's inside the cell, and they can sort out, so they're close enough to cell membrane, they can actually sense what's behind the membrane of the cell and the immediate environment. So here, if your external medium is, for example, ECM, right? What you do, and this red compartment represents integrins, okay? Then they sort out to be in contact with where you have the ECM. If your, for example, your green cells are cadherins, okay, that make homotypic additions between different cells. If you have 2 cells that come into contact, those cadherins will localize to the interface between those 2 cells, okay? And then if you have heterotypic cadherin addition molecules like the Fs and Fs, okay, they do not make transmembrane additions between the cell, but they make additions or connections between of the other type of membrane. Then you can also model that. And then here you have at the interface, you don't have green and blue. What you have is green-blue and blue-green. Okay, so and that's something that happens, as you have seen during PCP signals, where you have the distal molecules, they like to make transmembrane connections with the proximal molecules of the next cell. Okay, so the plan now is to actually not have you code along, but to start from already pre-code materials. So I'll ask everyone to go to the student materials folder. and download the file 7.3.1.zip and check what's written there. Okay, so the Hayden nicely, kindly just add the link for the docs in the in the chat. Okay, if you already have your Google Doc open and you don't see the file, refresh your browser, and then you should be able to see it. As you download and open, please take a look at what is written inside there, and try to guess what is this simulation will do when you play it. So if you open the file, what should you be able to see? You see that you have a contact and a contact internal plugins there. And you see that the external contact energies are all set to 10, and the internal contact energies are all set to, well, CBRG are set to 0, and the within same cell types set to 2, right? And you can already tell that you have 4 cell types. You have C, blue, red, and green. C is more for cytoplasm, but is color white, sorry, yellow. And you see that, so that is the only thing of notice that you can see at the XML level. Yeah, it's yellow. Okay, let's pretend that it's a word and a color. Okay, and so yeah, so on XML there doesn't seem to be anything of notice. If you look at the uniform initializer at the very bottom of the XML, you may notice that you are starting with a single cell, right? And this cell is a cell of type C. Okay, so you start a single cell of just with no compartments, with only one cell. But if you play the simulation, you see that if you play the simulation, and hopefully it will play, you see that you have lots of compartments inside. And the reason that it happens that is because at the Python level, there is a code there that actually add the compartments inside your cell. Okay. So let's now ask you to all take a quick look at the Python level. Okay. So if you look at Python steppables, and you may see that at the start function of the steppable, you have the series of commands. Okay. And the purpose of the series of commands is to create the blue, the red, and the green compartments inside your cell. Okay, so let me walk you through how this works. So first, on the top here, you see a loop over all the cells of type C. So that should be easy to understand. And then you have a series of commands here. Then there's some kind of commands on top of each one of them. So here in the first set of commands, I have this self.new cell, and then the type of cell. And what is basically this doing is creating a new cell of type B, and I'm storing it in the variable B. Okay, so note that this variable B is a dummy variable that I just created, and it's different from the self.b, which actually stores the cell type name. Okay, in our simulation. And then I do the same thing for the red and the green. I'm creating a blue cell, a red cell, and a green cell. And when I use these commands here, I'm not saying that they exist in any place in the lattice. I'm just creating the cell, but the cell does not exist in the lattice yet. Can you create BRG with the wizard, or is that different? Yes, you can create with the wizard. So let me just grab the wizard here very quickly. So let me show you here. So I'm gonna move this on top of here. Can you see? So if I want to create a cell, I go to Compartmental Python, cell manipulation, and I can have the option here, create cell. Okay, so when I click create cell, okay, that's one way to create a cell. well, that gives a full command. That's more than you need. But what's important is that on this side here, right, you have basically the self new cell, self type name, and you're basically creating your cell, okay? And you can ask it, so you can write in my cell equals self new cell and then the type of cell. So you get it, that comment here. You see that when you select that, you actually gave out also this part. This is actually used to draw the cell in a specific coordinate of the lattice sites. So while you are here, you can do the following. You can first just create the cell like this, and then this cell field access the lattice sites of your simulation, and you can give coordinates like 10, 10, 0, okay? And basically what you're saying is that at site 10, 10, and 0 on Z, so this basically is the X coordinate, Y coordinate, and Z coordinate. You associate that with my cell, and you can use that to draw your cells in the lattice directly, okay? So you can directly draw your cells on the lattice instead of using informationalizer. Of course, this will only allow to draw 1 pixel, right? What you can do, if you want to have a square cell, you can do 10 to 20, or let's say 10 to 30, okay, equals to my cell. And that basically does is that this is the initial coordinate and this is the final coordinate in X. So basically this will draw a rectangular cell of base 10 lattice sites on the X and height of 20 lattice sites on the Y direction. And all of that will be occupied by your new cell. Okay, now I see that your question was a little bit more basic than what I said here. Yeah, so when I set up the simulation, I add those out cell types in the wizard and to self-generate the base code in XML. Yes, I did that. But this is okay, is already an excuse to introduce your other things that you can do in CompCell. So many times you want to, the uniform initializer and the bulk initializer are a little bit limited, okay? Sometimes you want to directly draw the cells in a very particular configuration in your simulation, and want to draw them in a way that's parameterizable, right? And then what you can do, you can make a lot of, you can use the Python level and call this kind of commands out multiple times to draw the cells in a very specific shape, okay? In your simulation, so you can do that directly on the Python level. So that's another way to initialize the cells by directly drawing them in the lattice. Okay. Okay, so basically what I'm doing here is I just create the cells, okay? And before I actually draw the cells in the lattice sites, what I do, I have this new set of commands here, which I've already seen before, which was basically I'm grabbing whatever cluster ID my C cell has and assign that to my newly created blue, red, and green cells. Okay, so I'll assign. So those 3 new cells that I just created will be compartments of a cluster, which also includes the C cell, C type of cell. Okay. Okay. Then, okay, so I create the cells. I tell them they will be belong all to the same cluster. Okay. And what I do here in the next set of commands is actually I draw these new cells inside my cell. So what I'm doing here, I have in this command here, pixel list equal self get cell pixel list, and I give the cell as an argument. And what's basically is doing is that is giving me a list of all the lattice sites that are occupied by my cell. Okay. So once I have the list of all those lattice sites, well, here are counting them pixel, right? But it should be lattice sites. Then I go over each one of those lattice sites, and I retrieve the X and the Y coordinates of each one of those lattice sites. Okay, so I have those coordinates, and then what I do is I make a random number. Okay, I basically make a random choice between 1, 2, 3, 4, 5, 6. Actually, I think in your code maybe up to 10, right? So I select this random number, and then I check if this random number is 1, 2, or 3. And if this random number is 1, I assign that lattice site coordinate to belong to the blue cell. If it's 2, I assign it to belong to the red cell. And if 3, I assign them to belong to the green cell. And if it's something else, I don't do anything. I just let it stay as the C cell that I have to begin with. So basically what I'm doing here is I'm drawing these new cells that I just create as inside the original cell that I had, and I draw them in at random positions to not have any kind of bias in where they will be inside the cell. So it can be, they should be evenly distributed inside the cell, and they'll sort out according to the environment. So I'm creating a cell with multiple compartments without telling exactly where those compartments will be inside the cell. Does this make sense? Okay. So after drawing these compartments, can you do this with initializer? No, you cannot. There's no way to do it with initializer, at least for now. Yeah, there's no way to do it initializer. Maybe if it is start being more and more used by users, we may add a kind of a plugin that draw the cells with random distributed compartments inside. Okay, but for now we have to do from the Python level. Yes, you can provide different ratios of the internal compartments using this method here. The B, R, and G right now are just kind of dummy variables, so I can easily access those cells during this loop iteration. Okay, so if you have 2 cells, you'll be able to pixelize both of them. You first pixelize one, and then you do the second iteration of the loop. I grab the second cell, and then I pixelize that other one. Okay, so I draw my pixels there, and then the last thing I have to do, I have to assign volumes to all those compartments, right? So basically, I set the target volume of my cell, which is of type C, the blue, the red, and green compartments to be whatever volume they end up having after this pixelization, right? And then I assign also a lambda volume to all of those cells, compartments. So basically, that's what it's doing. Okay. So the reason that I share the code is because I didn't want to type all of this. Okay, and it's easier. So now, if you go and play the simulation, you don't play the simulation directly. Well, you can play directly and tell me what you see, and you will not see a pixelized cell. What you see is something that maybe look a little bit like this, okay? But if you stop simulation and play again, click on the step function, you see clearly a square cell to begin with all the pixels randomly distributed inside. And then you see those pixels quickly assembling and sorting out between themselves. So let me know if they are first, if the simulation works, and if you are able to see what I'm showing here, and also see the initial pixelized cell when you actually hit to the steps. So can you see that? So let me know in the chat. Thank you, Shalai. Yes, thank you. Okay, so people are sharing the screen. So yeah, you should see a cell that looks like this, okay? And then that it quickly becomes something that looks like that. So the question now that I have for you is that if you look at the energy of the internal energy of those cells, you see that they're all set to 0, okay? But that's not really enough to make all the cells stay mixed inside, okay? I want to, your first task in the simulation is, okay, what you have to change in it. Yeah, so that's your first task. Okay, what you have to change to make sure that those cells do not stay like this and to actually force them to be mixed like that. No, Sharon, is not because of low lambda, is because of another feature. So I'll give you guys a minute to play with the additional energies to make sure that the cell stays like that in a mixed state. I guess they are already all 0 to begin with. Oh yeah, oh no, I set them to 2. Okay, yeah. So yeah, so internal energy was not set to 0. Yes, correct. So well, there's no point playing this one because you don't have cells, different cluster. You only have cells that belong to the same cluster, right? You have only a single cell, so you have to change those energies. So what you have to do in order to get from here to here? Yes, I'm asking you why they do not sort. But yes, what you're describing is exactly correct. A few clusters disappear and some clusters just kind of grow and dominate. And then all the red pixels are there, then all those blue pixels are the other place, and all the green pixels are in this other place. Okay, Darshan suggests something to make them negative. Okay, try make the negative and tell me what you see, what you get. It feels like cheating, but it's okay. That's exactly what you have to do. Okay. So see, if you have a very low contact energy, that promotes the maximization of the surface between those cell types. That's true, but that's not really enough to mix them. Okay, so maybe you think that set those energies to 0 would be enough because now they don't see the energy anymore. And that induce some type of mixing, but not very strong, as you may see. It's not, it seems that it's not enough. You see that still have some kind of separation and clusterization to compartments. And the energy between lattice sites belong to the same domain, or let's say belong to the other different domains are the same, 0. So you feel like that should be no energy. So they should all mix. But the problem here is that the way that you grow compartments, right, or domains inside the pot's structure is by copying a domain to the next one or the neighbor. So because of the way that you grow is by copy or copy over one left side to a left side that's nearby, you have always a correlation between where your lattice sites are. So you have this spatial correlation of the lattice sites belong to the same cell due to the way that you actually create new lattice sites of a given compartment or a given cell. So the way to actually force the whole thing to mix is by giving actually negative additional energies to those compartments, because that then will actively promote the mixing between compartments. Okay, and any kind of if you have two fluids that mix together, okay, is because they have a negative surface tension between them. Okay, so in this sense, using a negative additional energy, it maybe feels like cheating, but it's not really cheating. Okay, molecules that mix in a fluid, okay, so you have a solution of molecules that mix, they have a negative surface tension between them. Okay, so they like to mix. Okay, so if you just make it so I am using those little j's for internal energies. Am I using big j's for the external energies? Okay, so that's just it's just a way that I like to use to refer to internal versus external. Okay, so if you all tried negative internal contact energy as -5, you should get something that looks like this. And I see that Melaine got it. So what value did you use, Melaine? -15, okay, very strong mixing. It's okay, right? All right, so if there are any questions or comments, then I can propose the second task of the thing, which is, okay, now you have all your compartments mixed, but let's say that the yellow represents a cytoplasm, so C for cytoplasm, okay? So if it's cytoplasm and all the other molecules are some kind of groups of molecules that would like to sit next to the membrane, okay? So what you want to do, probably you want to have a simulation to look more like this, okay? Now your challenge is to make this cytoplasm to stay at the center of the cell. So when you want to do this, what you have to do? So for this one, you actually have to do at least 2 changes in your code. You have to modify at least 2 parameters for this to happen. This is an ERP solution, which looks very similar to what is on the screen. Okay. This is Korean solution. Okay. Different cell colors, but she's getting it. So tell me, what did you have to change to get your results? That's right. That looks like a Christmas decoration. All right, so let me show the solution. So here, maybe what your first intuition. Okay, let's see the examples. Okay. Okay, so yes, Lampers actually made 3 modifications. Yes, Kirin have the solution that I also will suggest here on the screen. Yeah. So first, if you don't want your cytoplasm to contact the external medium, so maybe the first thing that you contact everybody minds, okay, let's just make the external energy, big J here between C and medium, to be very high. So here's at 40. But if I just do this, that's not enough, because then when you do this, okay, the C compartment does not touch the medium anymore, but you still, the other compartments really like to stay in the cytoplasm, okay? So maybe that maybe be a very good model depending on what biological process you are trying to recreate. So maybe you want those internal compartments to be freely diffusible inside the cytoplasm, okay? So that's not by at all undesirable thing that maybe even be more realistic in many cases. But if I want my simulation to look like this, I have to somehow prevent those compartments from mixing inside the cytoplasm. So the next thing you have to do, you have to slightly increase the contact energy between the red, green, and blue compartments with the cytoplasm. And you can either by making from -5 to 2 or from -5 to 0, or even from -5 to -1, that will already be enough to induce uh, uh, for them to actually stay more towards the edges than mix inside. Of course, if you adjust those little j's here, you can actually tune out how much of this internal mixing versus external segregation you have. And then you can choose those values according to your biological model, what you believe will be more realistic in your specific situation. But basically, to have this, you have to adjust those two elements, okay? So does this make sense? If you repeat those, if you copy those values here, can you get this result or not? So no, as you don't get this, or it does not make sense. Okay. Well, let me ask you first. So did you make the internal energies to be -5 to induce mixing? Yeah, okay, none of them. Okay. Now, if you make the external contact energy, so this is the external from cytoplasm with P40, so the cytoplasm have a really high energy with the medium, so you should stay inside. So you should avoid having any lead site that's touch the medium. So this very high energy here, you make all the yellow pixels to stay inside the cell, okay, and force the other ones to shield it from the medium. Okay, so this high external contact energy will induce this retreat of the cytoplasm pixels to the inside of the cell. Okay, but if you do that, because internally the cytoplasm still have a -5 energy with all the other cell colors, okay, you still have all these other lattice sites belong to the red, green, and blue, freely mixing inside the cytoplasm, inside the yellow cell, okay? So to prevent that, you have to slightly increase the contact energy between the yellow compartment with the red, green, and blue compartments. It's making my red cell behave the way yellow should be. Well, then check your cell colors in your Compartmental 3D player. They may be all mixed. They look right. So C is yellow, R is red, B is blue, and G is green. Okay. Well, then I do not know what's going on. So I'll just ask someone from the instructional team to get in touch with you, okay? Okay, all right. Okay, so the third task, okay, is I want you to suppose that you now have start from this situation here. Let's make all the red compartments sort out towards the medium. Okay. Yeah, so when I before I share the codes with you guys, I always save it and play that then a few times in the player, and that should, in principle, save whatever codes I use when I share the code with you. But what I'm hearing is that that does not always happen, apparently. Okay, so for this one, if you want now your red compartments to preferentially sort towards the medium, so let's say that the red compartments represent integrins that should, and the medium represents extracellular matrix, so it's a collection of fibers. So how you do that? RR internal energy does not play a role because you only have one R compartment inside your cell. So all the RR, blue, blue, green, green, and CC energies are not being used in simulation. They are zero. Okay. So there's no need to make any change to the RR. But what you said, Belen, about the increasing medium R contact energy, that's the trick. Yes. Julio, you almost got it. The only thing that's missing is that the blue and the green are segregated. They should be mixed to reproduce this figure here. Oh, great to hear, Sharon, that your colors are fixed now. Does everything make sense? Hopefully they do now with the red colors. Okay, Sheila, got it. Pete got it with the correct colors. So extra points to Pete. For this one, yes. Darshan got it. Okay, excellent. So I guess this last one was a little bit easier. So so basically what you have to do, you have to slightly increase. So no, sorry. All the external control energies are set to with medium are set to 10. So if you decrease the red with the medium from 10 to 2, that makes the red to actually go to the medium and also retreat from the medium, right? So then you have all the red compartments now sorting out towards the medium. Okay. So does this series of exercise make sense? I hope they do. And if you did not do the exercise, but you want to do the next ones, you can download the code 7.3.1b, and that have everything, all those, exactly those configurations implemented there, okay? All right, so with that, so this is everything that you can do for with a single cell. Now let's start playing with 2 cells, okay? So let's now to reproduce the patterns that you see here on panels B and D of the of this picture, okay? So your first task is not to play with any additional energy. You just leave them as they are. Your next task is to create 2 cells side by side. You can either do that on your own, or you can cheat and, well, you actually have to do that on your own. You want them to be actually in contact with each other, because what you want to do, you want to do these things here. So you when you create those 2 cells, they have to be either in contact or very close. Okay, Julie already gave the recipe to get it. Okay. Yeah, basically what you want is to have a initial configuration that, oops, that looks like this: 2 pixelated cells side by side. Okay, Sheila, I said that these cells look the cells looks very funny. Okay. Okay. So basically what you do, you just change the box max from 0 to, instead of from 20 to 44 on the X, you make 20 to 68, which you should be able to fit at least 2 cells of 24, okay, side by side. And when you do this, you start the cells pixelated, okay, but you may notice that as you evolve the simulation, it kind of looks like this, right? Which I'm not sure how to describe, although one of our colleagues suggest a few words to do it. I slice apple. Okay, that's nice. So, did everybody got a sliced apple as an output? Yes, yes. Okay. So if you got this, so you basically what you're doing is that instead of the way that I use the uniform initializer, I want my cells to be very big. So when you use cell compartments, especially dynamic compartments, one of the things that you have to do is you have to make your cells bigger. So that's the only drawback that you have in your simulations. Okay. So the cells are 24 by 24. Okay. And I use the box max such that just one cell would fit in my drawing box. Okay. And now if you want to have 2 cells, you modify your box sizes where you draw the cell so that actually you fit 2 cells this time. Okay. So if this, I have this. And now the second task, as after you have 2 cells, let's make sure that the cytoplasm or the C compartment are actually segregated. Keep the C compartment inside each cell. So now your next task, what you have to change the energies in order to get this output. Oh yeah, important note. You have to be the external energy. Okay, so try and show me the result simulation. And the question is will that be enough or not? Not enough. Okay. Sharon made a suggestion in the chat. you have an invisible cell. Do you really have an invisible cell, or do you have a black cell? Or it can be some kind of bug in the player, so try to close and restart it. Almost, Shilai, almost. Okay. I think you have to change a few other energies for your simulation to work. Yeah, actually, what Sharon suggested is the way to do. Okay, very good question by Villain. Is there a range of energy that you usually use for simulation, or it doesn't matter? I know that you use small numbers, but some simulation by other lectures use very large numbers. Okay, so to have this effect of inducing mixing between compartments, you have to either use zero or negative values. Okay, so that already gives a reference point. The other reference point that you can use is whatever temperature you set your simulation to be. If I'm using a temperature of 10, okay, then I try to have all my positive energies within additional energies to be at around 10, okay? So usually I set the energy of between two cell types of 10 to be the default, because that's also the temperature that I use on my, the noise level that I'm using in my simulation, okay? So then I like to set my standard contact energy to be equal to the temperature, whatever temperature value I'm using in the simulation, and then make things go a little bit higher or lower than that, okay? But of course, how much higher, how much lower, then it will be the zero will be the second reference point, because if I make things zero or below, I will start in this mixing, okay? So 0 and below will be mix-inducing effects, okay? Anything between that and 10 will be my normal range of energies, okay? And anything that gets higher than 10 or higher the temperature that I'm using simulation will start, is when I used to start induce a low affinity or actually a separation between the cells. If I actually drive the cells to really separate, then use a value that at least twice as bigger as the temperature. But that is the criteria that I use in my simulations, okay? I try to avoid using very, very, very high energies, because that might induce kind of pinning on the, well, your lattice sites start to kind of I'm start to be at risk of induced fragmentation. Okay. Okay, so at least that's what I do when I do computer simulations. I'm not sure that's the best way to do it. You have other experienced users here in this Zoom session. They can, if they want to unmute and tell what they use, they please guys go ahead and tell your answers. Okay, so I get here two results. This one from Lampros, it looks very good. This one from Schlide. Now Schlide has another one that now this looks better. Okay, so the way I did here is the solution that I have is actually Sharon had got it right. You have to not only increase the external contact energies of C with another C, but you also have to increase the external contact energy of C with everything else. So, for example, here in the example, I make everything 40, so it's a very high, 4 times higher than the temperature, to really induce separation and never allow a contact between them. Okay, so this is so I made those very, very high. Okay, so that is the trick. So if you use that, then you ensure that the cytoplasm stay at the center of each cell. Okay. You kept on training, got the same result. That's good. Probably is probably a better choice. Okay. All right. Now that you have here, I'll give you 2 tasks now, next. And this is going to be the final tasks, and then I'll end the module, okay? We didn't get to PCP yet, okay, but these are very important tasks to get you familiarized of how, so if you understand how to manipulate this small example, then you'll be able to really do the PCP exercise that we'll do later. So I'll give 2 more tasks, 2 more exercises. We'll do a break, and then when we come back, we'll do the PCP model to reproduce what have seen from Abhisha's work. And we also want to use the next model to do the do cell migration with anchors, okay, where the cell is migrating on a substrate, but instead of adding external pushing, use the external contact, external force plugin, or using chemotaxis. Let's do that by just have the cell make an anchor and pull itself forwards, okay, like this, okay? So that will be the plan for today. But now on to the next task. Once you have this working, now I want you to recreate the 2 following cases. On B, I do not want the red, I want the red to be mixed here, and I want the green to segregate between the 2 cells, okay? And on D, I want 2 things. I want the red to stay outside in contact with the medium, and I want the green to segregate towards the blue, and the blue segregates towards the green, but not green and nor blue. Okay, so these are the 2 outputs that I would like you to try. You can start with one, start with the other. I'll give you a few minutes, and then I'll share the answer, and we stop the module here and come back again for PCP and movement with anchors. Sorry, I was muted. Yes. So on the previous one, you can see that the blue and the green are randomly distributed on the interface with not, and so this blue here doesn't care if it has a green on the other side or there's a blue on the other side, and vice versa. Okay. On the challenge one that I have wants to do, you only, when you have green, you only have blue on the other side. So before, you have no preference coupling between green and blue. Okay. Here on the left, you have a coupling between green and green, and here on D, you have a coupling between green and blue. No, it was a good question. It was not completely clear from the pictures. Oops. I think Sharon just got bad luck. Yeah, once the cell sees the boundary, it automatically has, by default, zero energy with anything that is outside. We just don't have energy. So this happens. Yeah. Maybe just try it again. All right, so we have two solutions already. Julio got the D1, the B, and Darshan. Why? I cannot open the picture. Oh, sorry, that's Darshan's. Okay. And okay, and this is okay. So Julio got the B and Darshan got the D. Okay. No, Darshan, there's something missing here on yours. You don't have the red outside. So almost there. Eli almost got the D. I think he got it because where you have blue, you have the green and green blue. But I think whatever you did to induce this, it can be a little bit stronger. Okay, so it'll be more drastic, like you see here on this other one. Yeah, this one from Lampros looks good. The red's outside, and you can clearly see green, blue, blue, green, green, blue. Okay. Daisy got a B, very good. Looks exactly like we want. I see that in many of our simulations, you have boundary between everything, okay? What I like to do is when I run my simulations, I like to, okay. For example, I like showing a picture of the next slice. I like to have boundaries only between cells of different clusters, not between the cells themselves, okay? If you want to do something that looks like this, you can go to the visualization and unselect the cell borders and have only the cluster border selected. Okay, so that's a personal preference. When I do simulation of cells with compartments, I don't want to have any. I don't want to draw compartments between the cells, only between the clusters. Okay, so I always have the first one here to be unchecked, and only the cluster borders checked. or have those two boundary colors to be very different. So if you click here, you can set the cell boundary color to be like, for example, white, and the cluster boundary color to be black or some other color. So you can better tell wherever those two colors belong to the same cell, or do they belong to different cells. But that's just, of course, personal preferences. Okay, Julio now got it a perfect blue to green coupling at the cell boundaries. Okay. Sharon got a good simulation of the B case. Okay, and I can tell that she doesn't have draw boundaries between same compartments, only a white boundary between different clusters. You can clearly see the boundary between those two cell types here. So, yeah, very good. Yeah, it would be very nice if the Compartmental 3D player allowed to play with thickness of those boundaries to make it thicker than usual. So if you can make this thicker, it will be easier to visualize. Okay. So I think that most of you got it. Okay. So I'm just going to show the slide of the solutions. Okay. So for this one, I set the energy between medium and red back to 10, so that the cells will, all these compartments do not mind touch the medium, and set the energy between the external energy between green and green to 0, so they would like to preferentially go to this interface and have green on one side and green on the other side. For the D, what I did is first I reduce the energy between red and medium from 10 to 2. So the red preferentially goes where the medium is. And then to induce the heterotypic coupling between blue and green, okay, I reduce the external energy of blue and green from 10 to 0, while keeping all the other external energies at 10. So these are two recipes to get this output. You can make more or less drastic change in the energies, okay? But basically, that's how you can induce this or that kind of pattern in your cells, in your compartments. So the beauty of this is that if you increase the size of your simulation, you can add now 4 cells, 5 cells, 20 cells, right? And there you have no need to draw the cells by hand. Exactly, I'm going to put this compartment here, this here, this here, this here. You just add the compartment there at random places inside the cell. You make a simulation with 20 cells, right? And all those compartments you automatically sort out according to who is their neighbors and what the environment, its immediate environment is. Okay, so maybe that's something that you can do during the break. Increase the size simulation and add 4 cells. Okay, and then see that, well, either choose this one or maybe this one that's more rich in features. Make your simulation bigger and add 4 cells and see what happens. Then add 6 cells and see what happens, right? And you shall see patterns arising automatically because you're using dynamic compartments in your simulations. Okay. How about get the red cell to complete surround each of the two other cells? I think that's what's going on here. If I understand your question, no? Okay. Also between the two cells. Oh, so what you can do, you can make the, well, first, using this simulation as starting point, you go back or have the blue and green to 10, okay? And then what you can do, you can have, you can then make red and red to be 2. So I'll have this energy to be 2, and this one, red and red, to be 2. I think that will do the trick. Okay. Yeah, once you get a sense of how this dynamic compartments work, okay, then you can start playing with the energy to do whatever you want, okay? And there's absolutely no reason why you should only use 3 or 4 compartments, okay? You can use 10 compartments inside your cell, right? And make all sorts of rules. Well, the rules are implemented through manipulation of the additional energies, and then you get, okay, yeah, that's what I was talking about, right? Lampros just did a simulation of the case D with 4 cells, okay? That's what you get, right? Very nice. Okay, so I'll stop here for now. We did not do the exponential polarity, but we're gonna do after the break, okay? I promise, okay? And maybe when you look at this, you already can kind of guess what energy you'll be using, but I'll stop here and switch directly to the module 3 questionnaire,